All right, happy Wednesday. Any questions? We got a few people stopping by my office. Questions about the exam, about anything that's covered that I could address. Remember, exam Friday, homework due. Okay, all right. So don't be afraid. I got lots of office hours tomorrow. I'll be in my office all morning Friday. If you have any other questions that come up, email me. Today's lecture, including the little bit of Strept that we did last time, won't be covered on Friday. So, all right. So where we left off, we were talking about Streptococcus. So that's an organism those of you are in lab should be a little bit more familiar with, especially as we start our unknown today. In general, this organism's a little bit wimpier in the environment. It's a little bit harder to coax it to grow in the lab. We have to give it this special, really rich media, blood agar, so that it will grow. Inoculate that heavy. If you think you might have strep, inoculate that heavy. You still want to get isolated colonies when you're looking for your unknown, trying to identify your unknown, but inoculate that heavy. So we're zeroing in on strep pyogenes first. We are going to mention a couple of the other streps kind of briefly. We're going to spend a little bit more time on strep pyogenes and its virulence factors. So where we left off, we were talking about exoenzymes, and I think your book might talk about some other virulence factors. I picked some that especially may be related to something else that we've talked about or that were sort of easier to understand. Okay, so here's one, C5a peptidase. So as you're reviewing complement for the exam on Friday, remember C5a, it's usually referred to as a potent chemotactic factor, which remember, it calls white blood cells to the site of infection or injury. And when those other cells get there, right, they release cytokines that causes inflammation. So that chemotaxis and inflammation kind of go hand in hand. It neutralizes a potent chemotactic factor. Okay, so the enzyme that the strep pyogenes release, C5a peptidase, basically neutralizes that C5a. Okay, so less white blood cells coming to the site of the infection where the strep pyogenes is trying to establish itself. Streptokinase, that lyses blood clots. So we talked about our staff makes staphylokinase to lyse the clots. In this case, if there's an injury, for example, maybe some kind of wound, if the organism strep pyogenes can kind of navigate through those clots, it's going to be able to, again, establish itself. It's going to be able to invade deeper. Okay, so aids the spread of infection. Streptodornase, okay, this is kind of a fancy way. It's a kind of a DNase. So this, again, relates back to, if you remember, the three ways that neutrophils act. Okay, they're phagocytic cells. They will release their granules, their granulocytes. And that third way is by nets, the neutrophil extracellular traps. So if you remember, neutrophils have a way of kind of expunging their DNA in order to trap invading pathogens. Okay, well, here's a way that those pathogens get out of that. They can secrete the streptodornase that keeps them from being trapped. Okay, it breaks down that kind of sticky web of DNA, and so that they're not immobilized. A few exotoxins, streptolysin. So we mentioned before here, let me just back up a second to show you this picture, the different kinds of hemolysis patterns. Okay, so you could call the enzymes responsible for those hemolysis factors hemolysins, or in this case, when it's a streptococcus making them, we call them streptolysins. Okay, so if you're in lab, we talked about, your instructor probably talked about hemolysin O and hemolysin S, okay, or streptolysin O and streptolysin S. They just, act in different conditions to cause beta hemolysis. So a little bit more in depth about streptolysins. They not only act on the red blood cells in blood agar, for instance, to lyse them, they actually form pores in those red blood cells so that they lyse. It can also act on white blood cells and platelets, the other formed elements in the blood. And again, you can read a long time about this. I went down a rabbit hole earlier this morning trying to understand this better. But these tend to have a little bit of a different effect in vitro in the lab than if you try to understand their effect in an organism in vivo. And they don't seem to have quite the same effect in vivo. But let's see, what was I reading about that? It doesn't seem like I was reading the expression of certain streptolysins. When they are expressed in mice, it doesn't seem to alter the survival rate of mice, when they're infected with this, as to whether the organism is secreting streptolysins or not. But we do know that when they do lyse those red blood cells, that's just a source of a lot of nutrients for them. And when they act on white blood cells, it's a really kind of a dirty trick, because, in white blood cells, when they phagocytize, say it's a macrophage and it phagocytizes the strep pyogenes, it can secrete these streptolysins that cause the lysozyme to release its contents before it fuses with, the lysosome to release its contents before it fuses with the phagosome. So all those enzymes, they're now acting on the white blood cell and can bring about the death of the white blood cell rather than just killing what it's phagocytized. So kind of a dirty trick. Another exotoxin that's pretty rare, but again, this brings into a couple of things that we've talked about before, is this erythrogenic toxin. It's considered a pyrogenic toxin, and that means fever inducing. Okay, so strep pyogenes, I don't think I mentioned this before, this pyo, that means pus inducing. It's a very commonly misspelled organism. A lot of students want to put an R in there. So pyo means pus forming. It also, so we'll talk about when it forms pus in a minute, but also can induce fever. So this pyrogenic toxin, again, it's only in certain strains of strep pyogenes that can produce this, and we think that those strains are much less common now. But this toxin is a superantigen. So we talked about toxic shock syndrome toxin as being a superantigen. Remember, non-specific, lots of T cells activated, lots of cytokines, lots of inflammation. And the instructions to make this toxin is carried on a lysogenic phage. So if you remember, some bacteriophage can actually carry a gene from another cell in its virus and then actually change the phenotype of the new cell that it infects. Okay, that we call that lysogenic conversion. So we think only the strep pyogenes that have been infected with the lysogenic phage have the ability to make this erythrogenic toxin. And it's an important toxin. It causes a bright red rash all over the body. And that's a disease that we know, the scarlet fever. So we'll look at that in just a minute when we look at the different diseases. Okay. All right, so different kinds of infections. We'll start with skin infections and then get to kind of get some other infections as well. So we talked about impetigo, how that can be caused by Staph aureus, also can be caused by strep pyogenes. Looks very similar. Sometimes the strep pyogenes impetigo is described as being a little bit crustier, but again, usually will stay localized. Now, your book talks about how when impetigo is caused by strep pyogenes, it has a little bit more potential to kind of get in the bloodstream and cause a bigger problem, but that typically doesn't happen. Okay, so again, this childhood, kids pass it around, again, common in kids, need antibiotics so that doesn't scar. Getting a little bit more serious, erysipelas. Skin is often described as having an orange peel consistency, so kind of tough, little pits in it, maybe. Okay, looks kind of bright red, raised red lesions. Patients typically have high fever, also associated with that, typically vomiting, fatigue. Okay. Often it will start out if you have a little bit of some kind of wound or a burn or some kind of lesion on the skin, and lots of different complications. This can be fairly mild and kind of superficial, or it has potential to be deadly. So it's one of those things, if it's suspected, you want to have it taken a look at by a doctor. Very similar to erysipelas, but usually is maybe thought to invade a little bit deeper in the skin, is infection called cellulitis. And this can be caused by other organisms besides strep pyogenes. Once again, similar to erysipelas, can usually starts out by having a little break in the skin somewhere or a burn. GAS, that stands for Group A Streptococcus, and strep pyogenes. Usually some extremity, hands, face, legs, but you can see this hand is the one that's infected. It's swelled up, it's quite red. Okay, so localized inflammation, again, but usually, has the potential to go a little bit deeper. Fever, so again, can spread to the bloodstream and become life-threatening. So yeah, have that checked out if you suspect either one of those things. Okay. Getting then to another infection, it's not really a skin infection. It involves the layers below the skin, referred to as the fascia. That's why it's called necrotizing fasciitis. Necrosis just means death of tissue. Okay, so again, that connective tissue kind of below the skin and kind of what's holding all the organs together, that's the kind of deeper tissue that's infected here. It can start out from a break in the skin, but it's not always clear how patients get this. It's very treatable if you catch it early on. The problem is early on, it doesn't seem like it's significant. It can be, a little redness of the skin. It might hurt a little bit. Maybe the patient has a little bit of a fever. And then due to the exotoxins that are produced, proteases, lipases, things like that, that are just really rapidly eating up the tissue, it spreads really quickly. We would, of course, treat the patient with antibiotics, but because of the tissue damage, there's not good blood flow to these areas, so the antibiotics have trouble getting to where they need to go. Really high mortality rate. Usually surgeons have to try to keep ahead of this and just remove the infected tissue to keep it from spreading. Okay. And patients can develop, some other complications like toxic shock syndrome. So again, that's the worst one. Okay. Fairly rare, but it's out there. Talk to students that have seen it, dealt with it. Maybe some of you have too. Okay, so beyond kind of skin infections, the infection that I think of first when I think of strep pyogenes is strep throat. So of course, not all sore throats are due to strep pyogenes. Most of the time, they're going to be viral. But if you do have a really seriously hurting throat, usually accompanied by some fever, good idea to have that checked out. If you don't want to pay for a doctor visit, you can go to Meyer and get a rapid strep test, and they will, if it's positive, they will give you the antibiotics. So if you're somebody, especially that seems to get strep throat, that might be the way to go. Those rapid strep tests aren't really accurate. There's some false negatives and false positives associated with it. But yeah, question. Yeah, so what happens if you have your tonsils out? Do you not get streps more often? That's certainly been the case with me. I had my tonsils out when I was 35, and I've only had it once since then. I think that's the idea that sometimes the strep, if you have tonsils that are larger or really cratered, that organism can kind of just gain a foothold a little bit easier in those tonsils. So yeah, I can't give you numbers on that, but that's usually the goal when somebody has their tonsils out, to get strep less often. What does strep throat look like? Other signs and symptoms. Well, because of a toxin, a pyogenic toxin, that's one that we didn't talk about, but a pus-forming toxin. We often, or I would say sometimes, we'll see these pus pockets on the tonsils or in the back of the throat. That's not always there though, so you can't rely on that, like looking in your throat. Oh, it's not pussy, so it must not be strep. Don't rely on that. Usually have a fever, always have a sore throat, especially in kids. They can have a little bit more nausea and vomiting associated with it, which sometimes will kind of throw you off and think, oh, it's not strep because they're throwing up. Well, sometimes it is. How do we get this? Usually close contact, those large droplets, coughing, sneezing near somebody that has it, or maybe is about to get it, so you don't know that they have it. Swollen lymph nodes are usually present, and the patient is kind of, there's some secondary infections that are more common after you have strep throat. So things like bronchitis, pneumonia, and very rarely, again, certain strains that can develop into scarlet fever. So it's usually after a patient would have strep for a few days, and it doesn't get treated, and then bam, the patient breaks out in this bright red rash. My daughter, when she was 5, before she was verbal and could tell us, my throat really hurts, we knew she was sick because she had a fever, and then she broke out in this red rash, and it was a good sign, like, yes, we need to take you to the doctor now. And she did have scarlet fever, kind of freaked out the physician. I don't think he'd seen it very much. Again, it's not very common. But in the days before antibiotics, my husband's mother, she's in the 30s, she remembers her brother coming down with scarlet fever. The house was quarantined for 6 weeks. So, before we could treat this, there was some mortality associated with this. Helen Keller had scarlet fever, which caused her to become blind and deaf. Again, back in 1800s, this was more common and associated with some mortality. So this was a really serious infection. Now that we have antibiotics and we treat our strep right away, we don't see this as often. That erythrogenic toxin causes the capillaries near the skin to dilate, and that's where we get this really screaming red rash. It's not itchy or anything. It just is a sign that the patient is really, really sick. And sometimes this strawberry tongue, the bright red tongue, is associated as well. Okay, so again, strep throat is not one of those infections that you want to just ride out and wait till it gets better. You don't want to just gargle with vinegar or whatever. You want to go to the doctor because you don't want to take a chance that it will develop into scarlet fever. And there's some other complications of strep throat if you don't treat it. Some autoimmune complications like rheumatic fever and some, what we'll get to the next slide, glomerulonephritis. All right, so we talked about how strep pyogenes has that M protein on the surface. It's a feature so that it is less likely to get phagocytized, an anti-phagocytic factor. There's a couple different varieties of that M protein. If it's the rheumatogenic M protein, then if you have a patient that's ill with that strain, and again, usually with strep throat, and it's not treated, or maybe the patient only takes antibiotics for a couple days and then stops for some reason, typically a number of weeks after that, then the autoimmune complication, the rheumatic fever, can surface. And that's when we produce antibodies to that M protein, and it cross-reacts and targets things like the heart valve, especially the mitral valve. Okay, so apparently this is more common in kids. You get this strange looking rash very commonly. The nodules under the skin, apparently that only shows up in like 1 or 2% of cases. So I couldn't find a good picture of that. But this the patient can end up with some, chronic heart damage. Okay, sometimes it will heal up, but sometimes not. So it's estimated that between 1 and 3% of strep throat infections would progress to rheumatic fever if it wasn't treated. So in the days before antibiotics, 1, 2, 3 people out of 100 would get this. So you would know somebody, you would probably know a few people that had this, and then, maybe had a weaker heart, some, damage to their heart valves as a result of it. So right now we have very low resistance. Streptococcus has a very low resistance to antibiotics. So it's kind of the one organism where we don't have problems with resistance. If that should change and we start having some strep infections that don't respond to antibiotics, then we're going to see a lot more of this, and probably more scarlet fever as well. Okay, that other M protein, the nephratogenic M protein, we see a little different complication, acute glomerulonephritis. And without getting into the physiology or anatomy of the kidney, basically these antibodies are going to target and get parts of the kidney and get kind of stopped up in there. So the blood flow gets obstructed, blood pressure goes way up, urine output goes way down, and the damage to the kidney can potentially be permanent. I've known a few people that have ended up in the hospital with this. So we had an instructor that used to teach here. The 2 times that he got strep and the times that I knew him while he taught here, both times, even though he treated it and got on antibiotics, he ended up in the emergency room because his kidneys were starting to shut down. So luckily, his damage wasn't permanent, but again, you just gotta treat those strep infections with respect. What do we do? How do we treat either the rheumatic fever or glomerulonephritis? Well, you're going to treat with antibiotics just in case the strep pyogenes is not totally resolved. But the antibiotics are not going to help with the autoimmune complications. So there are drugs that we can give that will suppress the immune system, immunosuppressive drugs, and hope that we can kind of tamp down that antibody production so that the damage isn't worse. Okay. So that's pretty much what I had to say about strep pyogenes. I have less to say about the other streps. Strep agalactiae, that's in those Lancefield group B. Remember the Lancefield groupings are based on some carbohydrate surface antigens that most streps have, not all of them. This is known to form a capsule. And what's special about that capsule is that it's one of those T-independent antigens. So remember, we had two ways that we talked about how B cells get activated. T-dependent antigens require stimulation from activated T helper cells. T-independent antigens are recognized directly by B cells, and then the B cell, just from that interaction, gets activated to differentiate into plasma cells and make antibodies. So, you know, kind of in our response to T-independent antigens, we talked about, well, this is a really fast response. It's not as good a response because we only get IgM. There's no memory. And the big problem with this is we said that young children don't have a very good T-independent antigen response. So they don't really make very many or very good antibodies to these kind of antigens that are often parts of capsules of bacteria. So when babies contract a strep agalactiae infection, it's pretty severe. So things like pneumonia, sepsis, meningitis, pretty severe infections. We probably have some strep agalactiae, or it's estimated about 30% of us as just our normal microbiota. So women, usually at the end, I think, of their second trimester, are tested for group B strep, which strep agalactiae is the only group B. And if they're found to be a carrier, then they're given antibiotics so that during birth, babies don't pick that up. So good prenatal counseling or good prenatal care, and we can really make the incidence go way down for those kind of newborn infections. One alpha hemolytic strep that I'd like to talk about is strep pneumoniae. Remember, most of the streps will chain together. Strep pneumoniae tends to a little bit more often just make diplococci, so pairs. They also form a really beefy capsule. Remember, that's the organism that Frederick Griffith did his transformation experiments on, that strains that couldn't make a capsule didn't make mice sick at all. The strains that made the capsule usually caused the mice to die. And as you would guess from the name of this organism, one of the most common things that it causes is pneumonia. We're going to kind of skip over any virulence factors. This organism does have some specific ones, but we'll kind of skip over that, I think. So pneumonia, inflammation of the lungs. There's lots of organisms that can cause pneumonia, but this is generally the most common cause of bacterial pneumonia. And this organism used to be known as pneumococcus. So we changed the name, I think, I don't know, back in the 80s maybe, to Streptococcus pneumonia instead of the genus being pneumococcus. This is typically can be diagnosed by just abnormal breathing sounds, crackles, pops, a lot of coughing by the patients, really loose, phlegmy coughing, sometimes blood in their sputum that they cough up. Patients experience cyanosis, so they're getting a little less blood flow, less oxygen to their body, so their fingertips might turn a little bit blue, especially kind of underneath their eyes, a spot that gets kind of bluish. It's pretty treatable. This responds pretty well to antibiotics. Oftentimes, maybe patients are given a couple of different antibiotics because it is a pretty serious infection, just to make sure it works. This organism can also cause ear infections. Otitis media. Okay, especially common in young children because their ear canal is a lot more narrow and fluid tends to just not drain as well. So it just makes more ideal conditions for an infection. Very common for ear infections to come up after any viral infection. Okay, question? So what's the difference between walking pneumonia and real pneumonia? I think that walking pneumonia is usually caused by a different organism, a mycoplasma. That's a kind of bacteria that actually doesn't make cell walls. That's what's special about it. But I think it's not usually strep pneumonia. The walking pneumonia is a little bit less serious, but that's a good question. Okay. Sinus infections sometimes are caused by strep pneumonia as well. Another very serious, what we would call an invasive strep pneumonia infection, meningitis. Okay, so we talked about, how Staph aureus can cause meningitis. Now we have strep pneumoniae. We'll talk about a few gram-negative organisms that can cause bacterial meningitis, or one at least, coming up maybe next week or the week after. There's not a lot of organisms that can cross that blood-brain barrier. Again, it is very serious when they do. Again, a lot of inflammation associated with this. A pretty high mortality rate. Patients that do survive are often left with some kind of chronic disability, retardation, blindness, deafness. So definitely something we want to avoid at all costs. So luckily, we have a vaccine for strep pneumonia, which has made meningitis and pneumonia and even ear infections a lot less common. So sometimes we refer to this as the pneumococcal vaccine. This is one of those, remember we talked about a lot of different kinds of vaccines. This is a conjugate vaccine. So it's taking a conjugate vaccine, remember, it takes a weak antigen and conjugates or attaches it to something that's going to be more antigenic and more easily noticed. So those capsular, with this vaccine, we take the polysaccharides from the capsule, which again, those are the weak antigens. Usually polysaccharides, lots of repeating subunits, not as easily noticed, not complex enough to be noticed. And conjugated with a protein from the surface of the organism that causes diphtheria. So surface protein. It's Corynebacterium diphtheriae, but the organism that causes diphtheria, that's as a carrier. Okay, so we call that the brand name Prevnar 23. So that's one that you see advertised every once in a while. It's got 23 different varieties of, it sounds like ketchup or something, 23 different varieties of that different polysaccharide that we see on different strains of strep pneumonia. So with this vaccine, they started, this was introduced in about the year 2000, kind of an earlier permutation. They called it pneumococcal vaccine 7. The earliest permutation only had 7 different varieties. But when that was introduced, we cut the incidence about in half of invasive pneumococcal diseases like pneumonia and meningitis. And we've been, Prevnar PCV 13, Prevnar 13, that was a little better version of it, cutting incidence even lower. So now with the Prevnar 23 different varieties, we're hoping to get it down even lower. So again, this is a vaccine that's given to young babies. We want to keep them from getting those infections. Also older adults, there's a different formulation for people like adults of your age and my age that might be a little bit more susceptible to bacterial infections, like they don't have a spleen or whatever. But okay. So that's what I have on Streptococcus. Any questions, comments? All right. Sorry that I didn't get this posted till yesterday. I'm trying to stay ahead of y'all. Hopefully I'll catch up a little bit this weekend, get a little bit further ahead of you. So let's move on to, we did the gram-positive cocci, staph and strep. Let's move on to a couple of interesting gram-positive bacilli. So gram-positive bacilli, we mostly think of Bacillus and Clostridium as being the most common ones, the two major genera. Okay, so if we think about Bacillus, they are typically, we call them environmentals. So that means they can be associated with humans and with animals, but we mostly think about them as being pretty much any habitable place in nature, like the soil, water, pretty much everywhere you can think of. So they're everywhere. Most bacillus are not associated with any kind of human disease. There's a couple notable, especially one notable exception that we'll get to. They are aerobes. So they're catalase positive. And that's how we tell the difference, an easy way to tell the difference between bacillus and Clostridium, because the Clostridium are catalase negative. All right, so we don't use that catalase test just for staph and strep. We use it for other organisms as well. Some are even a source of antibiotics. So they secrete antibiotics. And another really famous thing about them is that they are endospore producers. So that's the bacillus and the Clostridium. Again, they're famous for making endospores. So they're going to form those in the environment. Okay, so when conditions aren't quite right, for instance, they will sporulate. Some are going to also form capsules, but they're not going to make capsules in the environment. They're going to express that capsule if they are in a host. Okay, so as a general rule, organisms that can form spores, they won't do that in a host, because when they're in the host, they're in a nice warm place with lots of nutrients. So they're not going to sporulate. They're going to do that in the environment when they're kind of hunkering down, right? And then again, they're not going to be encapsulated in the environment. They're going to be that in the host. Okay. When you grow bacillus on a plate, it has a very characteristic kind of wrinkly edge, as well as kind of a wrinkly appearance. They're kind of big colonies. So sometimes we get those as contaminants on plates, but we have worked with Bacillus subtilis in the lab a few times. So that's an example of one that's again not pathogenic. Sometimes we use that organism when we spore stain. So remember that Schaefer-Fulton spore stain, those of you in lab, the vegetative cells are red, and then the spores are these small green ovals. All right, so again, Bacillus subtilis, that's one that's not pathogenic. Bacillus cereus or B. cereus causes food poisoning. It contaminates whatever food, not a particularly serious form of food poisoning. And then the big one that we're going to spend a little more time on here is Bacillus anthracis, the causative agent of anthrax. Okay. So let's look a little bit more closely at Bacillus anthracis. Anthrax, it's very, very rare. So we think, well, why are we even talking about it? Well, it's just really interesting. And there is the potential. This is, of course, the weapon of choice of bioterrorists. From 1976 to 2001, there were only 25 cases of anthrax reported in the US. So about one a year, very rare. And then, of course, in 2001, we had the terrorist attacks of 9/11. And in that, we had 22 cases from that. 5 people died. So, the thought is, well, we kind of need to be aware in case something like this happens again. So again, fairly rare in nature. A reservoir, the soil is a reservoir, again, in that spore form. It's more common in some areas of the world. So more common to find spores in the soil in the Middle East, for example. In the US, again, some like North Dakota, apparently it's more common for those spores to be in the soil. So this is typically a disease of farm animals, of hoofed animals. So it's zoonotic. We only get anthrax typically, or traditionally, people have only got anthrax if they have been in contact with an animal that has it. So it's zoonotic. It's not considered a communicable disease, meaning people don't give it to people. They could if they tried really hard. I'll tell it that way. But as far as I know, there hasn't been a case of a person getting anthrax from another person. Okay, so let me just write that. People don't spread to other people. Okay, we get it from being in close association with animals. Bacillus anthracis has a very characteristic look when we stain it. It has these very boxy shaped, rod-shaped cells, which is that's kind of very specific to anthrax, and they also form these chains. So it has kind of a cool appearance. In terms of virulence factors, I'll only mention a couple. Strains that don't make a capsule cause very mild disease. So something about that capsule that makes it just help it survive when those spores germinate in hosts. It's a kind of unusual capsule that has a pretty high composition of protein. Okay, most capsules are polysaccharides, so that's kind of unusual. And I'm not sure what kind of capsule stain this is here, but again, you can kind of see the capsule here, again, secreted outside of the cell. It's going to make that, again, when it's in a host. It helps it resist complement. So that's going to just make it harder for our phagocytic cells to envelop it. The organism actually makes 3 different exotoxins. One I didn't mention because it's just a means to facilitate the entry of the other 2 toxins in the cell. So sometimes you talk about it as being a Trojan horse toxin. It just, again, kind of a sneaky way to get the other toxins inside the cell. And it's kind of nice. The toxins do like exactly what you think they would from the name. So the edema toxin causes swelling of tissues. Okay, so edema from inflammation. And lethal toxin, not surprisingly, causes cell death. What kind of cells? A lot of different kinds of cells, especially macrophages. Okay, so macrophages are a really important phagocytic cell processing and presenting antigen to get our adaptive immunity involved. So when the macrophages are dying, you're not getting a very good adaptive response to this. So, all right. So anthrax, it's a really good example of an infection that will manifest different ways depending on the portal of entry. Okay, so People get anthrax when they're exposed to the spores. And again, depending on how they're exposed, they'll have a different looking infection. So cutaneous anthrax, if you had to get a kind of anthrax, this is the kind that you would want to choose because it is the most common, but it's also the less likely to kill you. So this is when the spores are going to enter through a wound, through a break in the skin. And again, where would people encounter this? Usually when they're around animals. I think that's mostly the cases. So how does that present? That usually causes a nodule to form. A nodule is just an abnormal aggregate of cells. And when that scabs over, it turns black, okay, causes those tissue to die, necrosis of tissue. So that black scab that will form is, we've got a special name for that, it's called an eschar. Okay, so apparently it's not painful. It looks like it would be terribly painful, but most of the time this will heal up on its own and scar. But again, most of the time, again, people are fine. About 20% of the time, it will get the toxins will get in the bloodstream. And then it has the potential to be fatal. So, you know, about 20% mortality untreated. At the most, 20% mortality untreated. And usually, you know, that black scab, that's a little bit unusual. Hopefully, even though it's painless, people would seek treatment for that. Okay, and then, you know, with treatment, less than 1% mortality. So again, that's the kind to get. Who's at risk? Again, people that are closely associated working with animals, usually farm animals. In this country, areas of the country where the spores are more prevalent in the soil, we do have routine vaccination. We have a good vaccine for animals. So again, in this country, really low risk. And I think some of the people that 2011, of course, when the spores were mailed to Congress, who got anthrax, some of those people were mail workers that probably just were exposed maybe on their hands, and maybe it got through a cuticle in their finger, you know, that area that sometimes is a little bit potentially could find an opening. So some of those people had cutaneous anthrax. The other people and the all 5 people that died had pulmonary anthrax. So they got enough that those spores were aerosolized, they breathed them in. So this is the really one of the really deadly kinds of anthrax. Typically there's some kind of incubation period. Remember, you're not showing any symptoms in that incubation period. And then a prodromal phase, remember, mild symptoms, flu-like symptoms for maybe about 2 days. And then once, again, those spores have germinated and the vegetative cells are kicking out the toxins, then you get into the illness phase. That usually lasts only about 24 hours until a patient dies. So it's really fast acting, high fevers, pulmonary edema. So here's a chest x-ray. You get this mediastinal widening. So basically the center portion of the thoracic cavity looks much bigger than it should on a chest x-ray because of all the fluid accumulation, so much inflammation that patients go into shock. Again, when it gets to this point, it's going to kill you. And even if you have treatment during this invasive phase, then it's still a really high mortality. So you got to try to get treatment early on, either right after you've been exposed or when you're still in that prodromal period. Okay, we'll finish up anthrax and move on next Monday. See you Friday for the exam. Have a good day.